Hello, my name is Joshua Mugabi. I welcome you to Homeostasis. The term homeostasis is derived from two Greek words, homeo, which means unchanging or the same, and stasis, this means standing. Homeo means unchanging and stasis means standing. In its simplest form, it means staying the same. Staying the same. Let's look at the structure here. We have the ideal condition. In the middle, we have the ideal condition, and this is the normal condition where the system is supposed to be in the ideal condition. But in any case, the ideal condition can shift positively, moving upwards, or negatively, moving downwards. So if the condition increases, there will be a detection in the increase. And when increase is detected, there will be a response to lower the level to go back to the ideal condition, thereby maintaining the system constant. And if the system, the ideal condition, decreases, there will be a detection in the decrease from the ideal condition. And then there will be a response to raise the level back to the ideal condition. So the system will come back to the ideal condition or the normal condition. So that one is what we call staying the same, or what we call homeostasis. The system can be maintained at the same position. It is an organism's internal environment that stays the same. So it is the internal environment that we want to stay constant. For example, we can give an example of temperature. The internal environment of the organism, the temperature should remain constant. For the human being, it is 37 or 36.9. That temperature of the internal environment should stay the same, despite of the changes of the external environmental temperature. If the environmental temperature decreases, the internal temperature must remain the same. Even if the external temperature rises, the internal temperature should remain the same. So it is the internal temperature that must be maintained constant. The internal environment refers to the condition inside the organism. So whenever we are talking of the internal environment, an internal environment, we are referring to the condition inside the organism. We can be talking about temperature of the inside of the organism. We can be talking about glucose level, concentration in the environment, salt concentration, water. All those are internal environment. They are conditions occurring inside the organism. These conditions, they remain more or less constant despite the changes such as pH or temperature in the outside environment. So you find that you have the internal environment with the ideal condition, but outside it there is another environment outside, and the environment outside is likely to change, but when it changes it should not have an effect on the internal environment. The internal environment should remain constant. For example, if there is change in pH, there is change in temperature of the outside environment, the pH and the temperature of the internal environment conditions remain more or less constant despite of the change of the conditions outside of the environment. In practice, it comes down to providing a stable environment for the cells of the body. So by maintaining the internal environment constant, we are providing a stable environment in which the cells of the body are surviving. The cells are the units, functional unit, structure and functional unit of the body, and therefore they need a stable environment to carry out all the physiological processes of the body. So for that one to take place, they need a stable environment where the conditions are constant or the conditions are stable. The living processes in the cell depends on the activity of enzymes. So all those processes, for example, excretion, respiration, reproduction, all those living processes in the cells, they depend on the activities of the enzymes. And the enzymes, these ones, they work best in a stable environment with stable conditions. So the enzymes, they are the ones that speed up all the reactions that take place in the body, which constitute the living processes that are taking place in the cells. Therefore, these enzymes, they work best in specific conditions. So the conditions have to be maintained constant. For example, it might be temperature. The enzymes will work in specific temperatures. If it is pH, they will work at specific pH. So, meaning that they work at a stable environment condition. These enzymes, they work best in specific conditions, such as those of temperature and pH. So we are saying that these enzymes, if they are to help in the activities that are taking place in the cells, or if they are to speed up the activities that take place in the cell, they need specific conditions. And those specific conditions can be a constant temperature or a specific pH. So those environmental conditions must be maintained constant for the enzymes to work out well. Any change in these conditions affects the functions of the enzymes and may lead to the death of the cells or ultimately the whole organism. So when the conditions change and they move away from a particular condition, then the function of the enzymes is going to be affected. For example, let us look here. We are having the rate of reaction, for example, the velocity. This is the rate of reaction. And this one is the pH. So here we have our different enzymes: pepsin, sucrase, salivary amylase, agnase. Now, these conditions are specific for a particular enzyme. For example, let us look at enzyme salivary amylase. When you look at salivary amylase, it is showing us that it works best at the pH of 7. That's where it is working best. So which means that the condition inside, they should be at this pH. Any variation of the pH and the pH is lowered, we can see that when we lower the pH, also the rate of reaction is going to decrease. If we increase the pH from 7 and we take it a little bit higher, maybe up to 8, you can see that also the pH is going to affect the functioning of the enzyme. It is going to lower the rate of reaction. Yet if it was still at 7, you can see that at 7, the rate of reaction is at its maximum, meaning that enzyme salivary amylase works best when the conditions are maintained at the pH of 7. Should the pH of 7 be lowered, the rate of reaction is going to decrease. And should the pH of 7 be increased, then also the rate of reaction is also going to reduce, meaning that changing in condition is going to affect the function of the enzyme, and this one may lead to the death of the cell because of low rates of reactions. This is why the internal environment needs to be regulated and kept constant. So if there is any change, for example, in the environment of salivary amylase, the change of pH to 6 is going to affect the rate of reaction. Therefore, the pH has to be regulated and taken back to the pH of 7 for the enzyme salivary amylase to work best. In fact, the internal conditions are not absolutely constant, but allowed to vary within a very narrow limit. So we are not saying that the conditions will always remain ideal or the same. They can vary, but they are allowed to vary in a narrow limit, and within that narrow limit there can be a response to bring it back to the ideal condition or to the ideal environment. So it is not that the internal environment is absolutely constant. No, it will vary, but it is allowed to vary within a narrow limit which can be brought back to the normal conditions. Human body temperature, for example, varies between 36.1 to 37.8. So it is within that range. That is where it is functioning. We know that the temperature is 37 degrees, but it can go to 36.1 and then it can vary up to 37.8 within that narrow limit. That's where it is varying from. But we can't say that it is going to be 37 constant. No, it is going to vary within a narrow limit or a narrow range whereby it can be easily brought back to the normal condition. The average temperature is usually expressed as 36.8. So that is the average temperature, around 36.8, but we have seen that the environmental temperature can change, and when it changes, it will cause the internal temperature to vary within a narrow range, whereby it can vary from 36.1 up to 37.8, varying in that small range. But the average temperature is always expressed as 36.8 degrees Celsius. So, a better definition of homeostasis is the maintenance of the internal environment within a narrow limit. So that one we can now narrow down the homeostasis definition as the maintenance of the internal environment within a narrow limit, meaning that the internal environment is not always going to be ideal condition, but it is allowed to vary within a narrow limit, and in that narrow limit it can be allowed to go back and then taken back to the ideal condition, which is the normal condition. But we can't say that it is always ideal. No, it keeps on varying, but that variation is within a narrow limit. There is no specific organ which controls homeostasis, except possibly the brain. So homeostasis, the brain is the one that is mainly responsible for the control of the homeostasis, maintenance of the internal environment within a narrow limit. Now, when you look at our brain here, we have different parts of the brain. We have this big part which covers the biggest part, or this one here. That one is what we call the cerebrum. It is called the cerebrum. cerebrum. Then we have this hind part here. This hind part here, which is this part here, that one is what we call the cerebellum. Cerebellum. The upper one is cerebrum, the hind one is cerebellum. Now we have, from there, we have this part here. This part here is what we have here. This part here is what we are calling the corpus callosum. Corpus callosum. Now when we go to this side, we find a pinch here, or something like a branch here. That one is what we call the pituitary gland. It's what we call the pituitary gland. It is the same that we have here, our pituitary gland. Then on the upper side here of the pituitary gland, here on the upper side, as we are seeing here, we have what now we call the hypothalamus. Hypothalamus, which is the same. The pituitary is here. Pituitary is here, so the hypothalamus is here on the upper side. It's our hypothalamus. Now we have another major part here, the big swelling part here. This big swelling part here is what we refer to as the medulla oblongata. Now the medulla oblongata, now that one joins to the spinal cord. The spinal cord, which passes through the vertebra. So that is the part of our brain. We have the cerebrum. The most covered part is covered by the cerebrum. Then we have the point here, corpus callosum. Then from the corpus callosum, we go down here, we shall find the pituitary gland. And then above the pituitary gland, we have the hypothalamus. Then from the hypothalamus there, when you go down, you will find now the medulla oblongata, and then it joins to the spinal cord. And behind here, we have what we are calling the cerebellum. The cerebellum. So it is the brain that is concerned with the control of homeostasis. The skin, the kidneys, liver, endocrine system, nervous system, and sensory system, all of them, they play a part in the maintaining of the internal environment within a narrow limit. So all these organs, the skin, the kidney, the liver, endocrine system, nervous system, sensory system, they play a bigger part in the maintaining of the internal environment within a narrow range. Here we have our kidneys. We have the right kidney and the left kidney. They are one of the organs that are responsible for maintaining the internal environment within a narrow limit. The skin is a homeostatic organ which helps to maintain the body temperature within the limits, and that is around 36.1 up to 37.8. So that is our skin. If you look at our skin here, this is our skin. We have the upper surface. This upper surface, we call it the epidermis. Then we have the middle part, and this middle part here is the one that we term as the dermis. We have the lower part. This one is referred to as subcutaneous layer. It is mainly made up of fats. And we have down the blood vessels. These blood vessels, they are passing through the fat layer, and then we have the sweat glands, which form the sweat duct that moves up through the dermis and then through the epidermis to the sweat pore. So that is the structure of the skin. The sweat gland extracts sweat. The sweat gland extracts sweat from blood and passes it up the duct to the skin surface where it evaporates. So our sweat glands here, they are going to extract sweat. Sweat is extracted from blood. Is extracted from blood, and then it is passed through the sweat duct and it is taken to the surface of the skin. Now on the surface of the skin, it's where it's going to evaporate, and as it evaporates, it is going to take away the excess heat on the surface of the skin. And when this excess heat is taken away from the surface of the skin, now the skin is going to cool, therefore taking back the temperature within the narrow limit. The external temperature varies during the day, and it is from season to season. So the temperature keeps on changing season to season. Sometimes as much as 40 degrees Celsius. But the human body temperature stays at 37. So the external environment may change. The external environment may go above. For example, it may go to 40 degrees Celsius up, but the body temperature is going to be maintained at 37 degrees Celsius. It is maintained at 37 degrees Celsius, despite the fact that the external environment has changed to 40 degrees Celsius. The human body temperature stays at 37 degrees Celsius. This is achieved by sweating, vasodilation, vasoconstriction, and shivering. These are some of the mechanisms which are going to maintain the body temperature constant. There is sweating, the one we have seen, whereby blood is going to produce sweat, which is extracted by sweat glands and passed over to the surface through the sweat duct, where it is going to cause evaporation take place, and evaporation is going to end up cooling the body. Then we have vasodilation. Vasodilation, now that one takes place in the vessels, where the vessels that are coming towards the surface of the skin, they are going to dilate, and when they dilate, they increase in size, and they are going to cause a lot of blood to pass through those dilated blood vessels, and this will cause the blood to lose the excess heat to the external environment. Then there is vasoconstriction. Vasoconstriction, it is where now those blood vessels passing near the body surfaces, they will reduce in size, they will constrict, and this one means that less blood will be passing near the skin surface. Most of the blood will be passing on the lower surface, so that means that less heat is going to be lost from the blood vessels. That is when the external environment is lower than the internal environment. So much of the blood is going to be pulled away from the surface. It will bypass through the vessels that are passing underneath, but not coming on the surface. So there will be vasoconstriction. And then there is another mechanism, which is shivering. Shivering is whereby the body is going to keep on shaking, and as the muscles are contracting and relaxing, causing the body to shake, that one is going to be increasing the physiological process inside the body, where glucose is going to be broken up very fast. And when glucose is broken up very fast, we are going to get energy, which energy is going to supply heat to the body, and then the body temperature is going to rise to come back to the normal temperature. If the body temperature rises, the sweat glands in the skin are activated and secrete sweat onto the surface of the skin. So that is, if the body temperature rises, let us say it goes up to 40 degrees Celsius, and remember we want to maintain it at 37 degrees Celsius. So what is going to happen is that the sweat glands, they are going to be activated, and when they are activated, they will secrete the sweat, and when they secrete the sweat is going to come on the surface of the body. When it comes on the surface of the body, this sweat is going to be warmed up by the temperature of the body, and it will start to evaporate. As it is evaporating, it is carrying away the heat which was in the body, and as it carries the heat away from the body, that means that the body now is losing heat, and as the body loses heat, now the body temperature is going to change from 40 degrees to 37 degrees Celsius, which is the normal body temperature. So that's one of the mechanisms of cooling the body temperature and bringing it back to the normal. When the sweat evaporates, if the sweat evaporates, it takes heat from the body and cools it down. So we have seen that the sweat is going to be secreted on the surface of the body, and as it is secreted on the surface of the body, the sweat is a liquid. It is going to heat up, and as it heats up, because of the temperature which is coming from the body, it is going to start now evaporating, and it is evaporating. It is releasing the body which was in the heat temperature, which was in the body, is being released and carried out. So all this heat is going to go to the surface, and as this heat goes to the surface, it is going to be used to heat the sweat, and as it heats the sweat is going to evaporate, and as the sweat is evaporating, the temperature is reducing until when the body temperature goes back to the normal temperature of 37 degrees Celsius. Vasodilation: if the body temperature rises, the blood vessels in the skin dilate. That means they become wider and allow more blood to flow near the surface. So in vasodilation, what is going to happen is that these blood vessels are going to increase in size. These are the blood vessels that are passing near the skin surface. They will increase in size, and when they increase in size, it means that now they are going to be carrying a lot of blood, and a lot of blood is going to be carried near the skin surface. So that blood, when it is carried near the skin surface, that blood is going to lose heat to the skin surface, and as it loses heat to the skin surface, the heat is going to escape from the skin, and then it will go to the environment. That is because the blood vessels have become wider, and when they become wider, they allow a lot of blood to come to the surface, and when a lot of blood comes to the surface, it allows a lot of heat to disappear through the skin surface, and then it goes outside where it is going to evaporate. The heat loss from the blood through the skin helps to cool the circulating blood. So what is going to happen? This blood which was coming when it was very hot, as it is flowing, when it keeps on losing the heat, its temperature goes on reducing, and it becomes now cooler. When it is going out, it will be cold blood. The blood will have lost a lot of heat to the environment. Then we have vasoconstriction. Vasoconstriction. In vasoconstriction, that is if the body temperature falls. If the body temperature falls, then the blood vessels in the skin constrict. So what is going to happen is that these blood vessels, when the temperature falls down, the body temperature has gone down, then what is going to happen to the vessel? The vessels are going to constrict. They will become smaller, and when they become smaller, that means that a lot of blood is not going to come to the surface. So a lot of blood is going to pass through the underground or the underneath blood vessel. Less blood is going to be brought to the surface, and if less blood is brought to the surface, that means that the body now is not losing a lot of heat. Why? Because less blood is passing near the surface. Therefore, there will not be much loss of heat from the blood. The only blood that is passing near the surface is very small, and this blood is mainly to provide nutrient to the skin surface. And that one is brought about by the vasoconstriction. Vasoconstriction, these vessels that are coming near the blood, they become smaller. And becoming smaller means that they are going to allow very little blood to pass through them. Much of the blood is going to pass underneath the skin. And that one now passing underneath the skin is going to maintain the temperature of the body so that not a lot of heat is lost to the surface. Less warm blood flows near the surface, so less heat is lost. We have seen that when there is vasoconstriction, less blood is going to flow near the skin. And if there is less blood flowing near the skin, that means that there is less heat going to be lost to the environment. To carry out its living processes, every cell in the body needs a supply of oxygen and food. So we know that the cells are the ones that are carrying out the life processes. But these cells, when they are doing that work, they will need a supply of oxygen and food. So the blood vessels are going to bring in the blood, which is containing nutrients, and when these ones come, the cell can take the nutrients. They will get the nutrients and oxygen, and again the cells are also producing waste products. So now here they will now pass over the waste products to go into the blood. Waste products will be passed to go into the blood. The cells will give out waste products to the blood, and then the blood will take away the waste products. But for the cells to carry out the living processes, they need a supply of nutrients and oxygen. Waste products such as carbon dioxide have to be removed before they reach a harmful level and damage the cells. So when the nutrients have been brought and they have been taken up by the cells, and then also oxygen has been brought, it has been taken out from the blood to the cells are going to carry out the living processes. And during that process, they are going to produce waste products such as carbon dioxide. Now that one has to be carried away from the cell environment before it reaches a harmful level and it damages the cell. So it will be carried away by the blood vessels, and then it will be moved out of the body. The tissue fluid contains oxygen and food, which the cells can absorb and also accept carbon dioxide and other waste products produced by the cell. Now, when these nutrients are brought, they are brought by the blood vessel. And when they are brought in the blood vessels, they are going to be released into the tissue fluid. The tissue fluid is that fluid that surrounds the cell, is what we call the tissue fluid. So the nutrients and oxygen are going to be brought by the capillaries. And when the capillaries bring the nutrients and oxygen, they will release it into the tissue fluid. And it will be from the tissue fluid where it is going to be now absorbed and taken into the cells. So oxygen, glucose, they are going to enter into the cell, but coming from the tissue fluid. Then when they reach inside the cell, they are going to carry out life processes. And in the process, they are going to produce carbon dioxide. Then carbon dioxide will be released from the cells into the tissue fluid. And when it is released into the tissue fluid, then it will be carried and taken into the blood. And in the blood, it will be carried away from the cells before it reaches a harmful level. In cells, the chemical breakdown of proteins produces nitrogenous compounds like urea. So the cells, when they are carrying out their life processes or physiological processes, they will break down the proteins, and when they break down the proteins, they are going to produce nitrogenous compounds like urea. If these were allowed to accumulate in the cells, it would damage or kill them. So when urea is produced, it is going to be released into the tissue fluid because it is poisonous. And then from here, it will be carried into the blood vessels, which are going to remove it away from the cell environment so that it does not damage or kill the cells. The concentration of the blood and the tissue fluids tends to vary. So the concentration of the tissue fluid and the blood will keep on varying. How? Because remember, we are bringing in glucose and oxygen, and when those ones are brought, they will move out of the blood and enter into the tissue fluid, which means that now the concentration of glucose and the oxygen, as blood continues to flow, it goes on reducing. So meaning that the blood this side has less oxygen compared to the blood which is coming in. So there will be variation because most of the nutrients have been carried away. For example, if you look at this color, the color goes on changing the other side, meaning that there is variation in the concentration. Yet again, in the body, there has been carbon dioxide which has been produced. Now this carbon dioxide which has been produced is going to be allowed to enter into the blood vessel. And when it enters into the blood vessel, it means that now the blood is becoming more concentrated with carbon dioxide and less concentrated with the oxygen. and the tissue fluid is becoming more concentrated with glucose and oxygen and becoming less concentrated with carbon dioxide. So that's why we say that the concentration of the blood and tissue fluid tends to vary. It keeps on changing. It is not constant. It is important that the concentration of blood and tissue fluid is kept within a narrow limits if the cells are to function properly. So the concentration of the blood and also the tissue fluid should be maintained within a narrow limit if our cells are to work properly. So meaning that the content which is coming should be removed and the content that is being released should also be removed so that the cells can have a narrow limit through which they can function properly. The microscopic slice of the kidney tissue. So here we have the microscopic slice of the kidney tissue. We are going to look at the kidney and then try to see how it carries out its work to maintain the internal environment within a narrow limit. We have the blood pressure forces tissue fluid out of the clamp of the narrow capillaries. So here we have the blood. Blood is coming in this direction. As it comes, it goes into the glomerulus here. Now, because here we have the vessel is big, and when it changes into capillary, it becomes narrow. Means that here we have a lot of blood pressure, lot of blood coming. But when it comes here and the vessel becomes narrow, it means that the pressure inside that narrow capillary is going to be very high. And that blood pressure is going to push the tissue fluid out of the clamp of the narrow capillary. And when it is removed out of the capillary, it will enter into the Bowman's capsule. Bowman's capsule is that cup-shaped structure. The fluid contains urea, salts, it contains glucose and other soluble substances. So this fluid, which is forced out of the clamp of a narrow capillary, it contains urea, it contains salts, it contains glucose, it contains soluble substances. Now those ones, all of them, they are going to be forced to enter into the Bowman's capsule. The fluid trickles down the tubule and useful substances such as glucose are reabsorbed into the blood. So when all the content has been pushed into the Bowman's capsule, it will start flowing into the tubules. These tubules, we call them proximal convoluted tubuli, and then it comes down here, we call it descending loop of Henle, and here as it is going up, ascending loop of Henle, and then it goes to the distal convoluted tubuli, and then it provides to the collecting duct. So when the content has been released into the Bowman's capsule, it will flow through the tube. And as it is flowing through the tubes, useful substances like glucose are going to be reabsorbed to go back into the blood. This tubuli, more or less water is reabsorbed to maintain the blood concentration. So more or less water will be reabsorbed here in the ascending limb of Henle. More of water is going to be reabsorbed. The purpose of its reabsorption is to maintain the blood concentration constant. Excess water, salts, and urea collect here before passing to the bladder as urine. So we have the excess water which was not reabsorbed. We have salts that have not been reabsorbed. We have urea which has not been reabsorbed. All of it is going to collect into the collecting duct, and it will be passed to the pelvis. In the pelvis, it's where it is going to be collected before it is carried to the bladder, where it will be taken as urine. So now we go to the pancreas. The pancreas contains cells which produce digestive enzymes, but it also contains cells. Those are the alpha and beta cells. And these ones, they produce hormone, insulin, and the glucagon. So we have our pancreas. The pancreas has cells, and these cells, they produce enzymes. These cells, they produce enzymes. But again, it has got also cells which we call the alpha cells, and these alpha cells, they are the ones that produce insulin. Then it has also the beta cells. The beta cells are the ones now that produces the insulin, glucagon. They produce glucagon. If the glucose concentration rises, the beta cells release insulin. That is, if the glucose level rises, the beta cell are going to produce insulin. If the glucose concentration falls, then the alpha cells, these ones, they will release the hormone glucagon. And we know that glucagon, which is produced by the alpha cells, is responsible for breaking down stored glucose into energy. Or when the concentration of glucose rises, the beta cells will release insulin. insulin is going to cause the breakdown of glucose for it to be used up. And if the glucose concentration falls, the alpha cells are going to release glucagon hormone, which is going to break down the storage glycogen into glucose. Insulin stimulates the liver and muscles to remove the excess glucose from the blood and store it as glycogen. So we have the normal blood glucose level, which is around 90 milligrams per 100 cubic centimeters of blood. That is the normal. Now, what happens is that when the glucose level rises, it goes up. It is going to be detected by the beta cells. So when it rises, the beta cells are going to detect the rise in the glucose level. And these beta cells, which are in the pancreas, are going to release insulin. And insulin is going to cause the following physiological process to take place. One, it is going to instruct the liver and the muscles to increase on cellular respiration. Cellular respiration is going to break down the excess glucose to be used in form of energy. Insulin is also going to stimulate the liver to convert the excess glucose to glycogen. So it is insulin which is now instructing the liver and the muscle to convert the excess glucose to glycogen so that it is stored. And when it is stored, it will cause the level of the glucose in the blood to reduce. It is also going to cause the conversion of glucose to fats. All these measures are taken to make sure that we reduce the concentration of glucose in the blood. And then it will also cause the absorption of glucose into the cells so that excess glucose is removed from the blood. And when that one happens, now the glucose level in the blood is going to fall and it goes back to the normal glucose level of 90 milligrams per 100 cubic centimeters of blood. That is what we call the negative feedback. It has gone back to the normal because insulin has stimulated the liver and the muscle to remove the excess glucose from the blood and store it as glycogen. How did it do it? By increasing cellular respiration, by converting excess glucose to glycogen, by converting glucose to fats, and by also causing the cells to absorb the excess glucose. Now, glucagon This stimulates the liver to convert its stored glycogen into glucose, then released into the bloodstream. Now, glucagon is produced when the blood sugar reduces. We have the normal blood glucose level being 90 milligrams per 100 cubic centimeters of blood. So when the glucose level in the blood falls down, if this glucose level in the blood falls down, it means that now the alpha cells in the pancreas will detect that the glucose level in the blood has reduced. And these cells are going to produce a hormone glucagon. And glucagon is going to stimulate the liver to do the following. 1, it is going to instruct the liver to convert the glycogen which is stored to be converted into glucose. Secondly, it will also instruct the conversion of amino acids to glucose. And when all these ones are taking place, that means that the level of the glucose concentration in the blood is increasing. So this one will also cause the uncontrolled quantity of glucose enters from the intestine, entering into the blood. And when that one happens, then it is going to cause the blood glucose level to rise. And when it rises, it will go back to the normal blood glucose level, which is 90 milligram per 100 cubic centimeters of blood. That is a negative feedback. And then the blood glucose level goes back to normal. In this way, the concentration of glucose in the blood is kept within a narrow limit. So our concentration of glucose in the blood is going to be kept in a narrow limit of 90 milligrams per 100 cubic centimeters of blood by the negative feedback, bringing the level back to normal if it rises, and bringing the level back to normal if it falls down. If blood temperature rises, a heat gain center in the brain will send nerve impulses to the skin, which causes vasodilation and sweating, which cools the blood. So we are having the temperature of the blood is rising, and a heat gain center in the brain is going to send nerve impulses to the skin, which is going to cause vasodilation and sweating, which is going to cool the blood. So let us look here at our blood vessels. This is our skin, okay? This is our skin. And here we have blood coming in. This is the arteriole. It is dilated now. When it is dilated, we have the shunt vessels, the ones that passes below to pass over blood to the venue. They are going to constrict, and the arteriole is going to dilate, which means that a lot of blood is going to be passing through the arteriole to the capillaries, and less blood will pass through the shunt vessels and the connecting vessels. So much of the blood is going to pass via the surface of the skin. And when much of the blood is passing via the surface of the skin, it means that now it is going to release the heat. That heat is going to be released to the surface of the skin, and then it will be removed by the sweat, which is going to evaporate. This is because of vasodilation. Vasodilation, the arteries are dilated to allow more blood to pass near the body surface. And as the blood is passing near the body surface, it is going to lose its heat to the external environment. If the blood temperature falls, it stimulates the heat loss center in the brain, which sends impulses to the skin, causing vasoconstriction and cessation of sweating. Now, this is when the temperatures of the blood have fallen. The heat loss center in the brain will send impulses to the skin, which is going to cause the vessels to vasoconstrict and also stopping of sweating. So when we come back here, you will see that now the vessels, which are the shunt vessels and the connecting vessels, now they are going to dilate. When they dilate, they will be allowing much of the blood to pass underneath the skin. Now, the arteriole is now constricted. And if it is constricted, it means that less blood is going to be allowed to flow through the capillaries. And if there is less blood flowing through the capillaries, meaning that there is less heat which is going to be passed out to the surface of the skin. So a lot of blood is passing underneath the skin, which is causing blood to keep its temperature. And less blood passes near the surface of the skin, causing little loss of heat to the skin surface because of the vasoconstriction of the arterioles. These changes reduce heat loss from the skin. So we are seeing that the changes that are taking place because of vasoconstriction of arterioles, it is going to reduce heat loss from the skin. Why? because a lot of blood is passing through the dilated shunt and connecting vessels, which are passing beneath the skin. The warmer or cooler blood provides negative feedback to the thermoregulatory center in the brain. So if it is very warm or if the blood is cool, this is going to provide a negative feedback to the thermoregulatory center. The temperature regulatory center is located in the hypothalamus, a region of the brain just above the pituitary gland, which is closely associated with. So we have the hypothalamus. We saw that the hypothalamus is this point here above the pituitary gland. That point is the temperature regulatory center, where it is located in the hypothalamus. The hypothalamus exercises control of a number of regulatory systems. So the hypothalamus is responsible for many regulatory systems. Our hypothalamus is this part, which is above the pituitary gland here, and it connects to the medulla oblongata. It is the one that controls a number of regulatory systems. Another negative feedback is the control of the hormone estrogen. Control of the hormone estrogen. The pituitary gland produces the follicle stimulating hormone, or what we call FSH. This promotes the development of the ovarian follicle, and it also causes the ovaries to produce estrogen. So we have the pituitary gland, which is located in the brain. That one produces a hormone, follicle stimulating hormone. And when this hormone, the follicle stimulating hormone, is produced, it promotes the development of the ovarian follicles. And also it stimulates, or it causes the ovary to produce a hormone estrogen. The estrogen, of course, will go to the ovary, causes the repair of the ovary, and the estrogen still will be the one going to the brain to control the release of follicle stimulating hormone. Estrogen also acts on the uterus and causes its lining to thicken. So it is the estrogen which is going to cause the thickening of the uterine wall. Estrogen hormone, which has been produced by the ovary, is going to cause the thickening of the ovarian wall. When the estrogen reaches a certain level in blood, it acts on the pituitary and it stops it from production of more follicle stimulating hormone. So when the estrogen that has been produced reaches a certain level, it is now going to stop the pituitary gland from producing the follicle stimulating hormone. In this case, estrogen is exerting a negative feedback on the pituitary gland. Because estrogen has reached a certain level, it is the one now acting as a negative feedback to the pituitary gland, causing it to stop producing the follicle stimulating hormone. If estrogen level in the blood falls, the pituitary begins to secrete follicle stimulating hormone again. So if the level of estrogen reduces in the blood, then the pituitary gland is going to be stimulated to produce follicle stimulating hormone. Why? Because the level of estrogen has gone down. A follicle is a group of cells enclosing an ovum or the egg cell. The follicle, they are produced in the ovary, and it is a group of cells enclosing the ovum or the egg cell. The follicle enlarges and it protrudes from the surface of the ovary before bursting and releasing its ovum. So when the follicles are released from the ovary wall, they go on enlarging. That's the enlarge, excuse me. They will release the ovum out of the ovary. Hello, thank you very much for participating in homeostasis. Please do like, share, and subscribe. Thank you.